Let's continue our discussion on factors that are influencing enzyme activity. So here are some big ones listed: temperature, pH, the substrate concentration, any inhibitors that may be present. And here let's take also look at the issue of proteins that denature. If the conditions are bad, then first the enzyme, the rate of the reaction that it catalyzes goes down. But if it gets really bad, let's say the temperature is just too high, or the pH is too extreme, then these proteins, these enzymes, they will denature and then they don't work at all. Now, on the other hand, if the conditions are perfect, and substrate concentration is high, concentration of the enzyme is good, the enzyme has perfect conditions, well, then the enzyme will catalyze at its maximum rate. So here would be what it looks like when you're denaturing a protein. Think of denaturing. Picture an egg that you just put in a frying pan, and then you increase the temperature, and the egg changes its nature. You can literally see the change happening in front of you. And so on a molecular level, it's typically that this protein sort of unravels and becomes more, linearized. It loses definitely its three-dimensional shape. So this diagram gives you an idea of what it could look like for a particular enzyme, the optimal temperature at which it works. So this one looks like it works best right around body temperature for humans. So that's 37 degrees Celsius. So that would be right here. That's body temperature. And if it's cooler, it slows down about 10 degrees Celsius. You're pretty much down to 0. And then here, what happens as you're increasing the temperature, the activity of the enzyme will go down as well, because this enzyme will start to denature. Here's another example given of the optimal pH for some hypothetical enzyme. Here we have a pH optimum of about 5. And it will still grow at pH 4, and or just whatever enzyme, it will catalyze a reaction at pH 4 and pH 6. But if you go higher than 6 or lower than 4, the activity goes down. So this one here, this particular enzyme has a pH optimum of about 5. And here another example, given the role of the substrate concentration. Typically you are able to reach a maximum, and then increasing the concentration even more will not give you more enzymatic activity, because you have reached the peak rate. So once you're here, adding more substrate is not going to help. It's not going to increase the activity, because this enzyme is maxed out and it's operating at its highest rate. Now let's take a look at inhibitors. There are two types: competitive inhibitors and non-competitive inhibitors. So competitive inhibitors, they compete with the substrate for the same binding site, the same active site on the enzyme. So they're in direct competition with the substrate for the same spot on the enzyme. And this is what it could look like. So here we have the substrate wants to bind here to the active site of this enzyme. That's the normal substrate, and it fits perfectly in there, and it leads to a product formation. Now here we have a competitive inhibitor. This competitive inhibitor just takes the spot right there where normally the substrate would fit in, and now the substrate has lost its spot, and it can't bind to the enzyme, and of course then you're not going to get any product. Competitive inhibitors, they could be reversible or irreversible, depending on how tight they're going to bind to the enzyme. Here's an example given of what a competitive inhibitor might look like. You have over here para-aminobenzoic acid, PABA. That's a substance that's needed for microbial metabolism, and you can use sulfanilamide, a sulfur drug that is very similar in shape. The molecule is just very similar and fits in the same binding pocket as the para-aminobenzoic acid, and it will take the spot from the PABA and then not lead to the product that the cells need. So this would be a competitive inhibitor to PABA. Moving on to non-competitive inhibitors, They don't compete with the enzyme for the same binding spot, but they will bind to the enzyme at a different site. We call that allosteric site. So allo means other. So it's at a different spot, but they alter the shape of the enzyme, and so now the regular substrate doesn't fit anymore because the binding pocket has changed. And so that is known as allosteric inhibition or non-competitive inhibition. Let's take a look at a diagram, what this might look like. So here's the normal substrate, and it wants to bind here to this enzyme in the active site. Now here comes this non-competitive inhibitor and messes it all up. So it's binding right here, and it changes the shape of the active site, and now this the substrate, the regular substrate, doesn't fit anymore. So that would be a non-competitive inhibitor, and of course they can also be reversible or irreversible in their binding. Feedback inhibition is a great way to regulate enzyme activity and when you have a whole pathway. So a lot of times when you have enough of something, you would like to turn off the pathway because you don't need any more product. And so a very popular way in cells is that the end product, once it reaches a certain concentration, it inhibits an early player in an enzymatic or metabolic pathway, and thereby is shutting it down. So the end product of a pathway can become an inhibitor to an early player in a pathway, and that will be called feedback inhibition. And here you have such a pathway where the substrate is shown up there. Starting up here, the substrate binds to the first enzyme, and then we have another enzyme, another enzyme, and another enzyme. Eventually we're going to get an end product, and this end product starts to accumulate. Once we have a lot of end product, it starts binding to this enzyme 1 right here, and this will be an allosteric inhibition. So we have a non-competitive inhibitor right here, and it shuts down the pathway. Once the concentration of this end product is low, then it will come off of the enzyme returns to its regular shape, and the pathway will operate again. So it's a great way of regulating enzyme activity if you have whole pathways. Now, for the most part, enzymes are proteins, but there are a couple exceptions to that. Namely, RNA can function as a catalyst, and that is called a ribozyme. So that means RNA acts as an enzyme, and of course, RNA, it's a nucleotide-based molecule, so different structure from the proteins that are amino acid-based molecules. Okay, moving on to oxidation reduction reactions. A lot of times they are abbreviated as a redox reaction. So reduction, oxidation together. Redox reactions, they're paired. Reactions means one molecule gets oxidized and the other one gets reduced. The classic definition of oxidation would be the addition of oxygen. It's intuitive because we call it oxidation, so we're adding oxygen. A lot of times that's not what happens. Instead of adding oxygen, what usually happens is it's the removal of electrons or hydrogen in general. And reduction is the opposite. It means that we have either lost oxygen, that would be the intuitive way of defining this, but most of the time what happens is that we're gaining electrons or whole hydrogens. So one more time, oxidation, the removal of electrons and or hydrogen, and reduction would be the gain of electrons / hydrogen. Here you can see a diagram of a redox reaction. That means, so here you see substance A is oxidized because we're removing this electron right here. We're going to slap it onto B, and now B is going to have this electron, so B got reduced and A got oxidized. Okay, so in biological systems, most of the time we're not talking about the transfer of electrons alone, but we're talking about the transfer of hydrogens. Now, what's a hydrogen atom? It's just a proton, P+, surrounded by an electron, E-. So that basically would be one hydrogen atom, if you just want to do it like this: a proton in the center, that's the nucleus of this atom, and then a single electron balancing the proton right here. So this would be one hydrogen. Now, when we are removing a hydrogen, we are removing the electron and the proton, of course, and a lot of times in biological systems they are split. So that means the electrons again get processed separately, and the protons are also sort of gets processed separately. And you will see that when we talk about the electron transport chain and how that works. So in biological systems, oxidations are basically you're removing the entire hydrogen, so they are dehydrogenations, and reductions will be the addition of whole hydrogens with the proton and the electron. So here's an example of what this looks like in biological systems. Here you have this organic molecule and it has hydrogen on it. And then we're removing the hydrogen. In this case, we're removing 2 hydrogens. So that means 2 electrons and 2 protons right here. And they get transferred onto an electron carrier right here. And so now this organic molecule got oxidized because we removed the 2 hydrogens. And then here your electron carrier, the NADH, has received 2 electrons plus 1 proton, and then here's another proton here that can be used for hydrogen ion concentration gradients, for example, in an electron transport chain. Now, when we talk about energy metabolism, usually the goal is that we need ATP. ATP is the universal energy currency in cells, and cells can do a lot with ATP, but they have to somehow make it. Usually it involves the oxidation of food molecules that then ultimately are being broken down to CO2 and water, and we're gaining some chemical energy in form of ATP out of that. So now the question is, how is ATP generated? Well, it's a nucleotide-based molecule, so it's adenosine triphosphate. That means we have an adenine nucleotide, adenine base, and then we have 3 phosphates. And basically, cells they go back and forth between ADP, adenosine diphosphate, so 2 phosphates, and then adenosine triphosphate. So the difference between these two is this has a lot less energy than the ATP, and we can just phosphorylate, meaning adding an extra phosphate onto ADP to make it ATP. So let's take a look at this right here. So we have ADP, that's adenosine diphosphate, 2 phosphates right here. Well, we can put in some energy and slap on another phosphate, an inorganic phosphate. The I here stands for inorganic phosphate. And there we are. We have adenosine triphosphate with the 3 phosphates right there. Now, the generation of ATP can happen in mostly 2 ways. The most intuitive way would be substrate level phosphorylation. That means we're going to take a phosphate and add it onto ADP, and that generates ATP. So we'll just have some ADP as a starting molecule, and then we'll slap on that extra phosphate, and that generates ATP. That's substrate level phosphorylation. So right here, there we are. We have an extra phosphate. We're going to have ADP, and we're just going to slap that phosphate onto ADP and make ATP as a goal. The more efficient but probably less intuitive way of making ATP is called oxidative phosphorylation, where we're using an electron transport chain and chemiosmosis to generate a lot of ATP. And you will see that when we talk about electron transport chains a little bit later, how that actually works. It's not as easy to understand as the substrate level phosphorylation, but it's way more efficient and generates a lot more ATP as opposed to the substrate level phosphorylation. So here is a typical kind of electron transport chain where we start out ripping hydrogens off of food molecules, organic substances, and then they will be oxidized, and these hydrogens get slapped onto electron carriers such as NAD, and that makes them NADH or FADH2. And these hydrogens, they contain high energy electrons that then get passed on in this electron transport chain. So the flow of electrons, it's basically like when you're turning on some appliance, and the flow of electrons runs a motor, an engine, for example. And so here it's going to kind of same principle, where you're having a flow of electrons that powers these pumps, essentially, that then pump hydrogen ions on one side of a membrane. In the end, you're going to gain water right here, and typically a lot of ATP as well.